So for most of you, you heard me say this yesterday, but I'll say it again today. Thank you all very much for coming to learn about genome structure and its functional implications for biology. This meeting is the 5th, I think, in a series of meetings on this topic that started some number of years ago, before COVID, BC, in Varna on the Black Sea in Bulgaria, and has continued sort of alternating between Boston and Bulgaria for a number of years. It has been continuously sponsored by the National Science Foundation, in particular the program on the physics of living systems, and we have two representatives from that program here, Angel Garcia and Kristen Blagoyev, and we want to thank them for the continuing support, and they'd be happy to talk to you about all their research interests going on in the NSF, so find them at the break if you're interested in learning more about how the NSF is supporting this field. One thing I wanted to do is, of course, welcome you to Northeastern. As I discovered by just listening into conversations, not all of you are completely familiar with the vast amount of science being done at Northeastern, increasingly amount every year. In fact, you might notice that of the speakers to come in the next 3 days, 2 of them are actually from Northeastern. We have one group that does theoretical work on chromosome structure and one group that does experimental work, and you'll hear from people from those groups. But rather than me explaining that in detail, I'm going to give the microphone to our Dean of Science, Hazel, Sive, who came here to Northeastern about 5 years ago at this point, 4 years ago. During COVID, everything was a total blur. Who, after a long career in developmental biology at MIT, moved to Northeastern to be our Dean of Science, and I think she will explain to you a little bit more about all the exciting things going on here.
Want me to use that or the podium?
Whichever you prefer.
You know, I got notes. Hi, everyone. I have notes. That's one of the things you learn when you become dean, is that notes. I love speaking, extemporaneously, but actually, I'm less likely to say something incriminating if I use notes. So let me use notes, and let me say, before I forget, that I bought you all a gift, and it's right there next to Professor Levine, and I will make sure you each get this gift. You'll see what it is in a moment. So, good afternoon, everyone. I'm Hazel Sive, Dean of the Northeastern University College of Science, and I'm delighted to offer some opening remarks to this important NSF workshop on genome architecture and function. I'm really honored to welcome you to Northeastern University and delighted we're hosting this important meeting. I want to give warm congratulations to the organizing committee, including our university distinguished professor, Herbert Levine, for his leadership of the biophysics of living systems here at Northeastern and collaboratively beyond, and of course to other members of the organizing committee for bringing together this meeting, and many thanks to the NSF for their outstanding support of this initiative. So welcome to all of you, whether you are usually at Northeastern, whether you are here from across the world. It's wonderful to have you in this venue and to speak with you. In this short welcome, I'm going to do 2 things. First, I'll tell you a little about Northeastern University and the College of Science. And second, I'll give you a personal connection with the focus of this meeting. First, Northeastern. I call Northeastern the innovation capital of academia. This is a highly innovative university. It's also a global university comprising 14 different campuses across the United States and the world, with our nexus here in Boston. Our students are excellent. This year's undergraduate admission rate is 5%, and one attraction is our unique, empowering experiential education, where students can explore future career paths. Our signature program is called Co-op, and every undergraduate spends 2 or 3 6-month periods of paid work in research, industry, government, corporations, or organizations across the world. Co-op develops a can-do, responsible mindset in each student, a network of contacts and options for future careers. It's a terrific heritage from Northeastern's beginnings as a blue-collar school, retained and refined as we have developed into a top-rated R1 research university. The Northeastern College of Science has a unique vision to build what we call the good power of science in educating the next generation of science professionals and in solving the greatest research challenges of our time. Our research in the College of Science spans every scale, from mathematics to fundamental tiny particles in physics to molecules, cells, organs, the mind, large ecosystems, and the cosmos. Our outstanding research spans fundamental, curiosity-driven science to translational outcomes with emphasis on collaborative, high-impact approaches. Our wonderful physics department is no exception. In the 4 years since I've been Dean of Science, we've hired more than 50 tenure-track faculty members in the college, 14 in physics. About half of new physics faculty have joint appointments in other departments or colleges, emphasizing our landscape of cross-disciplinary convergent research. Biophysics is a major and eminent focus of the physics department, and we have many faculty in this area, from our most senior faculty, including Dr. Levine, to our newest junior faculty member, Dr. Maijia Liao. The overall group is defining a new College of Science Center for Theoretical Biophysics and is training PhD students in the area. Our faculty are building a new cross-disciplinary biophysics PhD program, part of the College of Science reinvention of PhD training from its kind of archaic past into the vibrant training that PhD training can be. To read out the good power of science, I've emphasized that its essential science researchers span all demographics of society. We are committed to being a diverse college where everyone belongs. I'm thrilled that since I've been dean, we've significantly diversified our new science faculty, including doubling the percentage of brilliant physics faculty who are women. Kudos to our physics faculty under the excellent leadership of Dr. Mark Williams, who will also give a research talk later in the program for this wonderful outcome. So let me move now to the second point of this introduction, my personal involvement with the topic. As dean, I have the wonderful opportunity to get invited to open all sorts of excellent meetings held here at Northeastern. In many cases, the field is way outside my expertise, but it's always interesting for me to try and bring a personal connection. For example, last month, I opened the Large Hadron Collider Physics Conference here at Northeastern and discussed how that group was defining particles that build the universe. And I contrasted this to my field of developmental biology that is defining how a person is built, and emphasized that discovery, the driving force behind brilliant science research, transcends and connects all our fields. However, today, as you will appreciate in a bit, I'm in the joyful position of connecting more fully with this important genome structure workshop. You see, long ago, as a graduate student in Bob Rader's lab at Rockefeller University, I wrote my thesis on cell cycle regulation of histone proteins. As you may or may not know, it is a fascinating aspect of the cell cycle that histone RNA and protein are cyclically synthesized, with a massive increase just before and during S phase, and a massive clearance right as DNA synthesis completes. My thesis addressed the question of mechanism underlying the cyclical change in histone synthesis. With Nat Heinz, I showed that both RNA synthesis and RNA processing or stability are tightly regulated. I identified the transcriptional activation of histone gene promoters through NF-kappa beta class regulators was involved, and I could reproduce that in cell cycle extracts. In retrospect, especially, the careful orchestration of histone gene expression to package DNA into nucleosomes at the right time is a stunning example of biological regulation. At the time of my thesis, however, understanding epigenetic genomic regulation was quite nascent. Nucleosomes were understood, of course, including that these needed to be loosened or removed during transcription. Histone modifications had been documented, but were largely unexplored. And to be honest, histones did not seem the most exciting proteins to me. How wrong one can be. You may know that the term epigenetics was coined in the 19th century and originally used to describe the events underlying the full development of a multicellular organism from a fertilized egg. Subsequently, in around the 1970s, the term became synonymous with stable and heritable changes in gene expression that cannot be explained by changes in DNA sequence, changing the meaning of the term epigenetics to the one that we know today. Presciently, for my postdoc, I'm not going to tell you everything that I've worked on, but I'll just tell you one more connected thing. Presciently, for my postdoc, I joined the group of Harold Weintraub, now of blessed memory, at the Fred Hutchinson Cancer Center in Seattle. Hal had a deep and brilliant engagement in epigenetics and chromatin regulation. At the time, the notion of 3D nuclear architecture was developing, but the tools available did not allow the in-depth, detailed analyses that have become possible and on which this meeting is based. I wanted to solve how the embryo is built and to identify the molecules behind the magic of development. This has been my life's research focus. I used the frog Xenopus, to which I'd been introduced as an undergraduate in South Africa, and which has helped open the fields of epigenetics and genome structure through both extracts and embryo analyses. From my postdoc work, I identified many genes involved in antero-posterior embryonic patterning and brain development, most of them transcription factors, with a good number of known chromatin modifiers. Subsequently, I joined the faculty of the Whitehead Institute for Biomedical Research and the MIT Biology Department, where I was on the faculty for 28 years before taking the honor and wonderful opportunity to lead the Northeastern University College of Science. Our research continues here at Northeastern, and it has opened many fields, including novel aspects of craniofacial development and into use of the zebrafish to address complex human genetic disorders. We have collaborated extensively on aspects of genome regulation. Most recently, we've identified chromatin modifiers amongst the set of key genes controlling a severe disorder mapping to human chromosome 16. So you see, when I pronounce that this meeting is very important, I have some qualifications to do so, and the absolute respect and excitement for the importance of your field. Congratulations on your accomplishments and your interests, and best wishes for an excellent meeting. Welcome to Northeastern University. And I'm going to put a little postscript in a box right next to where I was sitting is a gift for you all. At Northeastern University, those of you who are here know, but at Northeastern University, our mascot is the husky. You will see it everywhere. In the College of Science, we have adopted Science Husky as our official accepted logo. And I have Science Husky for you in that box there. Very compressed, but there is one for everyone. So I will leave those with Herbie or put them somewhere. Thanks very much.
So, Bill, it's a great pleasure to introduce you. Bill is a native of Massachusetts, grew up not far from here in the West Mass, and was a neighbor of Norman Rockwell. And here you see Bill featured on one of Norman Rockwell's paintings, The Right to Know. So this young gentleman with a few books in his right hand is actually Bill Earnshaw, whom you're going to hear next. That's Bill posing for Norman Rockwell. I found this picture somewhere. Bill continued after graduating from Lenox High School. He continued to Colby College in Maine, and then thanks to NSF fellowship as a graduate student at MIT in the King's Lab. During this time, I read that Bill took a class from Steve Harrison and Don Weinley, and I was like, wow, I also took a class from Steve Harrison and Don Weinley. And then worked with Steve Harrison, and that's his very nice Nature paper on the organization of DNA inside the head of a phage. And again, today you're going to hear about kind of bigger chromosomes than that. Then he continued. His first postdoc was with Klug, and the second one was Lemley. And that's this really seminal paper where chromosomal loops, the loops of mitotic chromosome, were visualized and characterized. And again, today you'll hear about what we understand about chromosomal loops in mitotic chromosomes many years since 1983. And also kind of fantastic collection of kind of 4 editions of the Cell Biology. For those of you who don't have this book, you can always order it on Amazon. The recent edition is 22, right? 2 years ago, a year ago? Came out about a year ago. I remember kind of reading the first edition of this one. And I know Bill through our Wednesday meetings for the last, I don't know how many years, 7, 8? Every Wednesday morning on the East Coast time and Wednesday afternoon, in Europe, we meet with Bill and Job and Anton Goloboroitko and others in our metaphase meeting. So this is most recent one. This is from 2021. And I kind of, occasionally we take snapshots when something really exciting is being discussed. So here you see Bill. This is 2020, very cheerful moment for some reason. I don't know why. That was in the middle of COVID, I guess. This is 2020. As you see, kind of we're getting younger as we go through this slides. This is also 2020. Bill is very much engaged. You see chromosomes on the screen. So that's kind of our weekly collaborations. 2019, before COVID. So we're even sitting with other people in the room. 18. So there are many memories in this snapshots. So as you see, this is before Zoom. This is Skype. There was such time before Zoom. This is a meeting in 2017 in Japan, where I drew a sketch of Bill. And so without further ado, I will give a podium to Bill, and we're really honored to hear about mitotic chromosome from someone who actually started working on this basically in the 1980s.
Yeah, so actually I have an interesting connection with the old days of Northeastern, because I don't know if it was in the tradition of the past of Northeastern, if it still happens, there were professional institutes here, and there was an institute for insurance agents who actually are required to take a series of exams to get the highest qualification. It was the Insurance Institute, and my father was the director here. All right, okay. So we're going to talk about approaching mitotic chromosome structure from all directions and from lots of different times. So here's the general structure of today's talk. I'll start by introducing chromosomes and chromosome scaffolds. I'll talk a little bit about some of the things we think we know about chromosome structure, and I've got something for the physicists and chemists to play with in there that's new, that they don't know about. And then I'll talk a little bit about what we're learning about mitotic chromosome formation. And I'm going to just give you a taster because Job's going to give you the serious talk. about that. So let's introduce chromosomes. So here we have Walter Fleming. I think I'll call it mitosis because of these thread-like structures. And this is, in my mind, the best movie of mitosis ever made by a guy named John Dahm in Oklahoma, Gary Gorpsky's lab. You see the whole process. Here we are prophase. The chromosomes are forming. The nuclear envelope breaks down. They line up. For the people who like signaling, notice that 2 chromosomes get lost over here. That one gets found, but this one, the cell waits. This is called the mitotic checkpoint or spindle assembly checkpoint. The cell waits for that. It's a very well characterized complex signaling pathway. And when that gets lined up, then you see this amazing event of anaphase. And if you see this, it either you go, yeah, okay, that's cool, or this gets under your skin and you can never think about anything else. So, here's the 140-year-old question. How do you get all that DNA in there? It's just an amazing problem. So actually, helical coiling of chromosomes was suggested all the way back in 1880 by Baranetsky. And here you see, it's clear. Chromosomes can be coiled. But maybe actually another popular model is that they don't really have a structure. They're just like spaghetti, or you could call it a polymer melt, just a jammed together bunch of nucleosomes. And there are a bunch of people who think that. And now I'm going to go off-piste. And I'm going to say, nobody can see, you can't really sort this out because nobody can see all the chromosomes, all the nucleosomes inside chromosomes yet in cells. But just wait, because it's coming. So my friend Ohad Medalia, our friend and collaborator Ohad Medalia and his team can now do this. So this is a thick section, a cryo-ET section, and these are individually modeled nucleosomes on what they see. So this is an actual map of where all the nucleosomes are near that region of the nuclear envelope. And when you do that, you can start to get views of how incredibly concentrated the nucleosomes are near the nuclear pores, and then there's a zone where they're less concentrated, and so on. So what our goal is now we're doing this in G2 chromosomes in a series of different cells. It's amazing how different the heterochromatin is in even in cultured cells. And then we're aiming towards doing this in mitotic chromosomes. But as you will see in a couple of minutes, mitotic chromosomes are a challenge beyond anything anybody thought. So now let's get back on track. So we had the helix, and then we had the nothing much, or maybe it's loops. And so you saw this picture. Job showed you this picture. I took it in when I was working with Uli Lemley, and we could see those loops. But the thing is, if there are loops, to make a loop, by definition, there's something holding the bottom of the loop. And so Uli suggested that there was something he wanted to call a protein scaffold, or a chromosome scaffold, at the bottom of the loop. So that would be a scaffold running along the chromosome. But then there was this huge fight. So, this is me, and this is the third referee. And it's not a joke, actually. There were any time you would just mention the words chromosome scaffold, your paper was rejected, because everybody knew that chromosome scaffolds were just an artifact of the methods used to make them. So I decided to do something different. At that time, I moved to Johns Hopkins to a cell biology department, and I discovered that people worked with antibodies. I'd heard of antibodies, but I'd never used them for anything. And so I decided to isolate chromosome scaffolds, make antibodies, and see if we could get evidence for an actual scaffold in cells. So what were the critical questions? So one of the things we needed to know is what's the protein composition of mitotic chromosomes? What are the scaffold proteins? Or another way of phrasing that would be, what are the critical proteins, not histones, the other things that you need to build a chromosome that looks like a chromosome? And if you can find those proteins, can you get evidence that they organize the structure of chromosomes? And to go all the way back to the big nasty fight, are they actually at the base of the loops? So, okay, so what are they made of? Well, this took us, those questions were 1980s questions. This is a 2010 question when we finally, with Yuri Rapsilber, developed the ability to do mass spectrometry, to isolate chromosomes. And so here you see, so I learned this back in 1980 and 81 in Uli Lemley's lab, how to isolate chromosomes. And then we put them into the mass spec. And it turned out to be an incredibly complex problem, which I don't have time to go into here. So this is the democracy plot. So one protein, one vote. As we go around here, red are things that we know are in chromosomes. You can see it's a tiny percentage of the proteins there. There's all this other stuff. And we call those proteins hitchhikers. And what we did was we developed a machine learning approach to figure out which proteins were actually chromosomal and which proteins were just sticking. As soon as the cell nucleus breaks down and the chromosomes go into the cytoplasm, a whole ton of proteins stick to them. Well, if you do anything you do to purify the chromosomes, those proteins are stuck to them, so they come along, but they don't have anything to do with chromosomes. All right. So if we take a more biochemical view of this, and if we look at mass, now you see, okay, half of the chromosome is histones. Well, we knew that, but we weren't really interested in histones because histones aren't in the scaffold. So what's in the scaffold? And those studies and other studies led us to study SMC proteins. These are proteins that are in the scaffold, and we'll talk about them quite a lot, as some of you may imagine. So we will return to the SMC proteins, but we're going to take a jump back in time again to how we tried to figure out what proteins were actually, so that's the proteins in chromosomes, but what proteins were in these chromosome scaffolds that people didn't believe really existed. So what are they made of? And the way to do that was to isolate chromosomes. Beautiful, this is a scanning EM by Adrian Sumner. Then you isolate the cells, you take the chromosomes. You need to get the cells, 90% of them in mitosis. This is not trivial. You isolate the chromosomes, and then you chew up the DNA and you extract most of the proteins, and you end up with things that you can sometimes, if you're lucky, they look like that. So it looks kind of like a chromosome, but it's got almost no DNA in it, and it's got some proteins in it. But what are the proteins? So what I did was, this is a story for the bar if the students want to know this. Let me just say that if you spend 8 months isolating a protein and you inject it into a guinea pig, it's not a bad idea to paint the guinea pig red. Because when we went to boost the guinea pigs, there was no red guinea pig in our cage after all of that work. And I found a red guinea pig in somebody, he had a red dot on the back of his head. I found the red guinea pig in somebody else's cage, and so I said, I'll be having you back. And that was Margaret Heck's PhD thesis. And so we got this band, there it was, and we did lots of chromosomes. We could see that the protein was labile and it had broken down. And we got immediate identification of this protein because a friend of mine had just purified the enzyme topo2, and lo and behold, if you used a topo2 antibody, that's what you had. So we had immediate, even with our first identification, we had a peptide map. We knew that we had topo2. So topo2 is a major scaffold protein. So now one of the questions we wanted to ask is the scaffold a rod along the axis, or is it something else? And so that was Margaret Heck. She did immunogold labeling of chromosomes, and this is not for the faint of heart, I can tell you. And you can see, well, what you see is a bunch of spots. But if we blow it up, you can see that each of these spots is a clump covered with gold. So each of these spots is a little local concentration of topo2. So the scaffold is not a rod. It's some sort of a diffuse network that you can see as a bunch of lumps scattered throughout the chromosomes. And here's another example of it. You can see topo2 is concentrated at centromeres. There's a centromere. It's more concentrated there. So the first conclusion we get is it's easy. There is no rod-like scaffold up the axis of chromosomes. There's a network of proteins that maybe organize chromosomes. We didn't know that yet. So are scaffold proteins actually important for mitotic chromosome structure? We couldn't answer that question with topo2, and still these days, yes, topo2 is important, but exactly its role in chromosome structure is still kind of interesting to study. So we isolated we got another protein. It was scaffold protein 2, which became SMC2, and there's SMC2 on the axis of the chromosomes. Now, SMC2 is a protein component that's required to make a complex called condensin that many of you have heard of and that we'll come back to in this. And 4 groups reported the discovery of these SMC proteins at the same time. So we now can do an experiment, or Kumiko Samajima can do an experiment where she rapidly and thoroughly depletes SMC2. We take cells, we synchronize the cells in G2, we release them into mitosis, and we do this in the presence and absence of SMC2, or condensin. So if SMC2 is there, you get nice chromosomes. If SMC2 is not there, you don't see anything that looks like a chromosome. But if you look at the diameter, that was not intended. Let's be irritating. All right, if you look at the diameter of this and the diameter of that, and remember that volume varies with the cube of the radius, then this is highly condensed chromatin. But it doesn't look like chromosomes. Okay, so we got some answers. So what's the protein composition? There's more than 4,000. Probably nowadays with DIA, there would be 6,000 or 8,000 proteins. There's a ton of proteins you find there, but mostly it's histones and irrelevant hitchhiker proteins. To build a chromosome, it's probably fewer than 20 proteins that are specialized in actually building that structure that we see. Plus, there's 100 more that make the kinetochore, but I'm talking about making the shape of the chromosome. And do they organize chromosome architecture? Yes, they do. I just showed you if you don't have them, you don't have chromosomes. But now, if we go all the way back to Uli Lemley's idea, are they localized at the base of the chromatin loops? Well, I'm going to jump in and out of the story here. So we'll go to SMC proteins, and I'll just get the introduction out of the way here. So condensin shapes mitotic chromosomes. Cohesin holds sister chromatids together, and it also is very important. It's a driving factor organizing the structure of the interphase, or the organization of the interphase chromosomes. Now, for those of you who don't work on this, it's slightly unfortunate that there are two condensin complexes, and what's unfortunate is that they were named in the order in which they were discovered, which has nothing to do with the order in which they act. So it turns out that condensin 2 is in the nucleus, and it's the first one that starts building mitotic chromosomes. Condensin 1 is in the cytoplasm, and there's some in the nucleus, but it mostly starts acting after nuclear envelope breakdown. So there are these two condensin complexes, and what do we think they do? Well, evidence from many labs, including some simulations here from Leonid's lab, show that condensin, we believe, worked by extruding loops of chromatin. That's here, and here we have a simulation by Anton on the right. Anton's simulation is going to show that if you let this run to completion, you basically make the two chromatids into bottle brushes and you separate them. And then Kees Dekker and now other people, many other people, have found conditions in vitro where they can get this loop extrusion to happen, and you can see it on a single molecule basis. So here you see this as the simulation runs. When it eventually ends up stopping and rotating, you'll see that we've actually, by just extruding loops, we've managed to separate the two chromatids. Okay. So if scaffold proteins can extrude loops, they should be, if you're extruding the loop, you're pushing it out, you should be at the base of the loop, where Lemley said they would be, not out in the loop. We haven't tested that for condensin, but we've done that with some experiments for topo 2, which I like to show, but just because I think they're so cool. And they'll be like nothing you've ever seen before. Well, so first of all, this is a picture that I took. This is one cell blown way up, swollen with hypotonic medium. That's the DNA. Doesn't look like anything if you can even see it in this light. If we now look at the topo 2, there's the topo 2, and all of a sudden we can see things that looks like chromatids there. So it looks like the topo 2 is concentrated on the axis of the chromosomes, but there's a lot of space out here, and if we had topo 2 molecules distributed all out there, you wouldn't see them. They wouldn't be bright enough. So how can we tell if topo 2 is where we can't see it? So what we're going to do is we're going to think about antibodies in a way which I would imagine none of you have ever thought about antibodies, and that is molecular ratchets. So what we can do is we can take chromosomes, and even though you know that histones are supposed to neutralize the negative charge on the DNA, there's not enough positive charge in the histones to do it. You have to have divalent cations in addition to the histones to actually neutralize the charge. So if you get rid of the divalent cations, but you leave the histones there, the whole thing swells up into a big ball, which you can then centrifuge onto an electron microscope grid, and it looks like a crepe. So now, antibodies, remember, antibodies are bivalent, and they don't let go under the conditions that we usually use. You can abuse antibodies terribly and get the antigen off of them, but the antibodies are usually dead at that point. So they're bivalent, and they usually don't let go. So antibody cross-linking is stronger than electrostatic repulsion. You don't just put antibodies in high salt and get the antigen off of them. So if we could find the right amount of antibody, it would cross-link its antigen within chromosomes. And so the idea of the experiment is this. You start with a swollen chromosome, which you assume is kind of just breathing in space. Now you put an antibody in there, and one arm of the antibody grabs, and that's stuck for the rest of this experiment. And then as the chromosome is breathing, eventually the other arm grabs, and now that's a cross-link. And if you think about what's going to happen, eventually the structure will just get trapped smaller and smaller and smaller. So it'll look, if the antigen is everywhere, when you put the antibody in, it'll look like that. And here's an example of doing that. This is anti-topoisomerase 1. So this chromosome should have looked like the one I showed you before, like the crepe or a pancake all spread out, but they don't because the antibody trapped it. So then Margaret Heck did this experiment. So if the antigen is not everywhere, but the antigen is only at the base of the loops, then what the antibody will do is it will cross-link the base of the loops, but the loops will stay out, because you're not pulling them in. Or you don't pull them in, but you're not trapping them when they're in. So Margaret Heck did that experiment, and here is a swollen chromosome in the buffer, and it doesn't look very interesting, it's just a big ball. Now she put pre-immune serum, and you can see some fat droplets from the serum, but you can see that it's just exactly the same size. Now she put anti-DNA, and that's what you get with anti-DNA. So the question is, what do you get with this anti-topo2 antibody that we made? And the answer is, it looks exactly like these guys, except if you can see it, there are two dark axes here. So topo2 is not stably bound out in the loop chromatin. It is right where Louis Lemley said it would be, at the base of the loops, and right where we would think it should be if it was like an SMC protein or analogous to an SMC protein that was extruding loops. Okay. All right, so the first scaffold protein forms a network through the chromosome and stably associates with the chromatin only at the base of the loops. So are they localized at the base of the loops? Yes, fine. Now, what we think we know, so the dogma is that SMC proteins build chromosomes by loop extrusion. I showed you the simulation. Question is this straightforward? So the simulation and the in vitro experiments are in vitro. What about in cells? Is this straightforward? Well, let's see. How compact actually is the chromatin in chromosomes? This is a little present for the physicists who may be surprised here. Some of them. Leonid won't be. So to figure out how concentrated things need to be, you need to know how many cubic microns, or how many nucleosomes is there per cubic microns. So you need a great microscopist, and that's Fernanda. And what she does is she uses serial block phase scanning electron microscopy. It's a way of getting serial sections of cells. I would be happy to go through this with the students if you want, but you can get sections of an entire cell and you never lose a section. And then if you have a program called Amira, you can recognize the chromosomes and distinguish them from the cytoplasm, and then you can build three-dimensional models of these. And the resolution in this microscope, or the microscope that we use, is 4 nanometers by 4 nanometers. And then the way it actually does the steps in Z is it uses a microtome and it slices off. And those are 60 nanometers. So it's 4 by 4 by 60 nanometers. And it's done by Alison Beckett and Ian Pryor in Liverpool. They actually run the microscope for us. So Fernanda did complete three-dimensional reconstructions of 14 RPE1 human cells. You see them at various times through mitosis here. And the first thing that we realized when we started, actually we realized this was one of these papers where all of the big insights come when you're writing the paper, not before when you're designing the experiments, and not even when you were going over the experiments. The first thing we realized was if we looked at 4 different cells at the total volume of chromatin in the mitotic chromosomes, it was incredibly constant, 140 plus or minus 5 cubic microns. Really, really constant. So that means that the chromosomes go to the chromatin packs to some kind of density, and it's at that density. Well, but it's not only metaphase. which I would have put my hand in the fire and said that that was the case at metaphase. But in fact, here you see pro-metaphase, the chromosomes aren't lined up yet, 150, and at anaphase, 145, another later anaphase, 142. So it's as though the chromatin reaches a very, very constant density. Well, is that density interesting? Well, we can do some good old-fashioned calculations. You know, the T, the golden path through human chromosomes, we had an extra chromosome in there, so this is the number of base pairs in our cells. That's the total volume of the chromosomes in metaphase, so that's how many megabases it is per cubic micron, and that's how many nucleosomes it is per cubic micron. Okay, it's a number. So that's not that cool until you just do this crazy calculation. So I never thought I would calculate the molarity of nucleosomes. The molarity of nucleosomes. 0.7 millimolar nucleosomes in mitotic chromosomes. This is insane, in my opinion. It's just, and to put this in perspective, if you isolate nucleosomes and you're an expert at phase separation in vitro, and you get the nucleosomes to phase separate in vitro, as Michael Rosen and Daniel Gerlich and their colleagues did, you get a nucleosome phase, and it's 342 micromolar. This is more than twice as concentrated as separated nucleosomes in a phase. So you see why I say that, well, OHAD is taking these amazing pictures of nucleosomes at the periphery of cells, but trying to see individual nucleosomes inside something this concentrated is going to really push the computational modeling. All right, so. And you know, this is what I would have expected, that the chromosomes would peak at metaphase. It's been published by others that they peak later, but this is concentration over various phases. But actually what it is they reach a plateau. It's not a peak. And that suggests to me that actually this concentration of chromosomes, that the final concentration of nucleosomes and chromatin is likely anyway to be some sort of a phase separation. Okay, now, does this have any implications? Well, SMC proteins, you saw, I showed very quickly, Case-Decker's experiment of SMC proteins extruding loops. Well, it must take a lot of effort to jam nucleosomes together at a concentration of, this is an old previous calculation, it's 760 micromolar. So are they strong enough? Well, actually, that's interesting. Condensins, here's a force versus rate. The world champion motor is FTSK. Phage T4 packaging motor is pretty good. But condensins 1 and 2, they're really fast. So this is the effort that we've measured in vivo, or that we've calculated in vivo from our high C measurements. But the stall force is around 1 piconewton. The stall force of a kinesin is 6 times stronger. That's a motor that moves things on microtube. So these are really weak motors. So they can extrude loops really quickly in vitro, and they have some amazing properties. But in vivo, are they really strong enough to jam stuff together that concentrated? I wonder. I think there may be much more interesting, a lot more interesting things to discover there. Okay, so the chromosome morphology is aberrant after SMC2 ablation, but chromatin compaction still occurs. I showed you that slide before. So if condensin's not compacting the chromosome, what is? We'll take 4 or 5 slides, I guess, to quickly show. Maybe it's not a motor. Maybe it's epigenetics. Maybe it's histones. So what Alyssa Zhitaneva did was she isolated histones with all their modifications intact, which you do by an insane protocol where you take cells and you drop the cells into concentrated sulfuric acid. It doesn't sound like very biological, but it works for histones. And so she made histone octamers, and then what she did with them is she used John Whittam's, an array from that John Whittam designed, and assembled phased nucleosomes on this array. And then she did that with interphase histones and mitotic histones. Same DNA, same conditions, just interface histones or mitotic histones. And Kazu Miyashima in Japan had developed this protocol where you could just take chromatin like that and put it on a slide. And what you see is, you don't see anything if you use just DNA or if you assemble the DNA with interface histones. But if you do it with mitotic histones, there are clumps on the slide. And if you look at this in the atomic force microscope, what you see, if you assemble interface chromatin or mitotic histones, you see the mitotic histones clump. So I think that the part of this, at least a good part of this compaction of the chromatin is driven by the pattern of histone modifications. And what would it be? Well, we collaborated with Ivan Matic, and what you see in green is what goes away in mitosis. And if you look at all these red things, they're all acetylations. So Alyssa and Ivan concluded that it's probably changing in acetylations of histones could be what drives mitotic chromosome compaction. Unfortunately, if any of you are mass spec people, you will know that two of the most difficult proteins to study are H2A and H2B. The peptides are the wrong size, they don't fly well, they're either too small or too large, so it's really hard to get more information on that, but we're trying. So where have we got so far? We resolved this 20-year-old controversy. Non-histone proteins have a really important role in chromosome structure, condense and shapes chromosomes, but it doesn't compact the chromatin. Histone pulse translational modifications may, in fact, be what compacts the chromatin. We don't know what the critical modifications are, but probably deacetylation of H2B is one very interesting candidate. And in fact, Michael Rosen and Daniel Gerlich showed that deacetylation of histones can, in fact, promote liquid-like phase separation, so that may be the case. So maybe phase separation is really important, but I think there's a lot more work that needs to be done on that. And then, in the last little bit of this, we'll talk about what we think we know. So if, here's Fleming, well, I want to study mitosis, but in a typical culture, there are 5% of the cells are doing mitosis. So, so who is this Kumiko Samajima character anyway? As some of you know, so here she is. You may not know that Kumiko's had to set up a team that ran Scotland coast to coast in the course of one night and has done other long distance. She started running when she was 49 years old, wanting to keep up with her kids. And she is now a serious long distance runner. Anyway, you've said hi already, say hi again. So here's what she did in our lab. And she just presented this in a group meeting without telling me she had been working on it. That's kind of the nature of our relationship anyway. Kumiko does what she wants. And this is chemical genetics. So you have a typical cell cycle. And what you do is, using Kevin Chocot's technology, you make an analog sensitive allele of CDK1. Now it turns out you can't do that with human CDK1, you can't do that with chicken CDK1, but you can do it with Xenopus CDK1 because the human and chicken are dead. Xenopus 1 is still alive. If you add this drug 1 in MPP1, it sits in this slightly larger binding pocket and completely kills the enzyme. And the lovely thing about that drug is you dilute it and it just washes right out. So what you do is you wash it out, and the cells enter mitosis synchronously. Now, this looks like art, but this is one of Caitlin's pictures. And basically what it is, this is an example of the student being a little bit sloppy and getting an getting an air bubble in her slide when she mounts it. So this is not an art form. This is an air bubble. And these are all perfect early prophase cells. You would never, ever see a population of prophase cells like this. They're all in prophase because when we use Kumiko's method, within 2 minutes of washing out the drug, all the cells are in prophase. Well, 96% of them are in prophase. So here's one of Kumiko's movies. These are chicken DT40 cells, and you'll notice that the chromosomes condense around the nuclear envelope, and the ones in the middle are condensing around the surface of the nucleolus. All right, so what we did with that, and this was in the early days of our collaboration with Job and Leonid, I met these guys at a meeting in the south of France, and I just wouldn't go away. I kept telling them we had to do this, and we had these, well, then they got fully on board, I have to say. So, all right, we could show that, that all the interface features went away. You had a chain of loops, and then this was definitely one of the, this was one of the, I don't think Johann said eureka, but it was kind of like this moment where we got this, holy crap, you would not believe what we just saw. And that's this second diagonal. And the reason why the second diagonal is interesting is that says that everywhere along the arm of that chromosome is interacting non-specifically with somewhere else 2 or 4 or 6 megabases up the chromosome. And the only way you could do that is if it's some kind of a helix. And so that was super exciting. So we published that, and that paper has been pretty successful. And right, and so we spent 7 years on a follow-up study working together. Kimiko made 19 new cell lines. These are AID and halotag knock-ins. We now can do mitotic synchrony at a 2.5-minute resolution. We do 3D reconstructions of the entire cells. We're using conventional and super resolution microscopy. Kimiko and Ataru, her husband, have designed a whole quantitative proteomics pipeline. And then Anton and Leonid, with the rest of us just sitting, well, Job actually knows what he's talking about. None of the rest of us know what we're talking about. We just watch Anton and Leonid talk about as they're the new ways of interpreting high C. So I hope you'll learn about, Job will tell you a bit more about that, and I'm going to offer a very brief sneak preview. So if we look at what the chromosomes look like, so this is Fernanda now not doing human cells, but looking at chicken cells. And you can see the same kind of thing that she saw with the human cells. So these are all cells 30 minutes after release into mitosis. So they're all actually in prometaphase. And if you look at the total volume, first of all, you can see these two look the same, except actually, if we rotated this, you would see all the sister chromatids are separated because this is cohesin has been knocked out here. They're all separated in there, whereas here they're all like single tubes. But look at the volume, 70 cubic microns, 68, 72. These are bigger, but they look like fuzzy things with a core on the inside, and the core is 72, really concentrated. And so lessons that we could learn, one very short lesson we learned from all of this, which one of the referees at Science is going to believe us, but he's having a hard time getting, or he or she are having a hard time getting their head around, is these guys are all cylinders, but without condensin 2, it's not a cylinder anymore. The chromosome becomes a random coil. Anyway, Job will probably take us there. And so, what can you learn? Here's the same region in interphase and in mitosis. The high C is just, it's clear that there's a ton of, and that's the centromere, and boy, what's going on there? Because this is not an artifact because we can't sequence the DNA, because there's the same region in interphase. All right, so, what we did in this study was we made chromosomes that had only cohesin, only condensin 1, only condensin 2 in various combinations. And I think our minds were blown from week to week. I mean, it's just amazing. So here example, if you have only condensin 2, you have amazing high C. You have a second diagonal, a third diagonal, a fourth diagonal. And the microscopy is ultra boring. And then if you have only condensin 1, you have this amazing chromosomes in the microscope, and the high C is amazingly boring. So, and it turns out that this was the hardest high C pattern to model for Anton. But Job may tell you about that. So the biologists couldn't interpret these data on our own. There are times when you really just do have to have a physicist. And and Anton is the in-house guy. And here you go, Job. Job, you might want to look at this one. There you are. Anyway, this is Anton presenting one of his newest models. Sorry, I just didn't want you to miss that. And so the sorts of things that you do when you're doing this modeling is you say we have complexes that are pumping out loops. How far apart are the complexes? Well, what do you see if they're 40 nanometers apart? They're not touching. For sure they're not touching. 40, 60, 80, 100 nanometers apart. It actually has implications for what the structure looks like. The further apart they are, the more scattered through the whole chromosome the scaffold will be. And I'll show only one example of this, and I'm pretty sure that Yoba will show you the two more interesting ones. And this just shows you that the blue line that you can't see here are the data, and the orange line that you see on top of it is the model fitting the data perfectly, calculated by Anton, and that's showing nested loops with these various parameters, which we don't need to go into. And the reason why you have the second diagonal going up is because these loops are long enough to reach up and touch the loop above them. So they have interactions up the axis of, it's not a regular helix, it's a very irregular, it's a sort of I think we call it a helical trend. It's a helical, it's not a nice, beautiful helix, but anyway. So you will learn more about that. I did not try to teach you about this in detail in this talk. So but maybe here's what they would have told you, so you can look for some of this in Job's talk. So I would have told you, they're rod-like cylinders with nested loops formed by a non-histone scaffold composed of SMC proteins. Interactions between cohesin and condensin make the scaffold, and they give those interactions give the chromosomes their cylindrical shape. Chromosomes lacking cohesin and condensin 2, that's the one, condensin 2 started in prophase, it makes big loops. They're no longer rod-like cylinders, they're random coils. Polymer simulations reveal that condensin can somehow bypass cohesin as it extrudes loops. The scaffold is this chain of SMC complexes that follows a disordered path. And polymer, one of the things I think is interesting and has implications for those of you who love high-resolution microscopy is polymer simulations tell us there is no one structure of a mitotic chromosome. There's no reproducible structure. There is a trend of a type of structure. So looking at individual examples by light or electron microscopy and saying this is what a chromosome is like are probably misleading. Okay, so I could have also shown you the speed at which they extrude loops, which is 1.5 kilobases a second. All right, so what are the big unanswered questions? So we still don't know. I mean, I still can't tell you, I can't draw a path for how the chromatin fiber is organized in chromosomes. The models show what we can see, but they're just models. Now we have atomic structures of condensins and cohesins and atomic structures of those guys with DNA on them, and we still don't, I think it's fair to say that we still don't know how loop extrusion works, or how they give chromosomes their cylindrical shape. And as far as I know, nobody's yet tackling this problem, which I find fascinating, which is here's one of Fernanda's images, and look where that chromosome is being formed. This is in late prophase. The chromosome is not flopping around in solution, randomly extruding loops in all directions. It's stuck on the inside of the nuclear envelope. And what does that do? That's, I think, a really interesting unexplored question. And the models that we have can't tell us why the chromosome follows that helical path. We don't know. So I've been studying chromosomes for over 40 years with more and more powerful techniques, and they're still hiding their secrets well. So what we need at this point is we need new ideas, we need new techniques, and I need like 10 more years. Okay, so an amazing collaboration. Kamiko, who I hope that all the students know, and many of the faculty either do know or will know, Fernanda, who is on maternity leave at the moment, Ataru, who's done a lot of the mass spectrometry, Johan is the high C Meister in Job's lab. Anton was Leonid's graduate student who went to become directly a group leader. Samir was a PhD student, is still here in Boston, probably making more money than the rest of us, I would guess. Anyway, having a good time in industry. I didn't have time to show you any of Samir's really amazing simulations. And I have to say that we couldn't have done the EM without Alison Beckett and Ian Pryor. And an old friend of mine, Jim Paulus and Linfeng Ji, who are both at the University of Wisconsin in Oshkosh teaching undergraduates, make 1 in MPP1 as a lab exercise with their undergraduates, and they give it to us. And so, yeah, here's my picture for the group. Yeah. And I would like to thank the Wellcome Trust for funding our work to the level which we would like, and here's the group. So thank you very much. Hope he's still alive.
Questions?
That was an amazing talk. Thanks a lot. Really enjoyed. I still puzzled. I think you addressed the point about these motors being so weak. Are they actually acting alone, or we have like multiple motors acting alone? Basically, we like to do these pictures that appears that you have extrusion on this very diluted medium with one chromosome pulling these things around, but we should maybe have a much more complex mechanism in terms of multiple motors actually doing the game. Do you have any idea about it? Want to comment?
What about the paper I'm looking at? I'm terrified because I'm sitting here in front of Job and Lee. It's not exactly. There's some papers that recently have come out in Science about cohesin, saying that in certain instances cohesin functions as a monomer, and in other cases it seems to function as a dimer. And I think those are using really sophisticated light microscopy to do the quantitations, looking at step bleaching, for example, and at FCS. So I would say for cohesin, maybe monomer, maybe dimer, but no evidence that I know of much higher order. And for condensin, I don't know. Do you know?
Don't have some traffic jams or condensers sometimes around or areas that they may be? Because amazing that what's these motors? They're all less than a piconewton, right? The stalling force, right? So it's like...
Yeah, okay. So there's of course the one protein that we absolutely don't understand, which is, in fact, it's sitting out there for those of you, as some of us near retirement age, the kinesin, KIF4A. This is a kinesin. It's a dimeric protein. It binds DNA. It's in the interphase nucleus. In neurons, it moves targets down, it moves proteins down the axons just like a good old kinesin. At the end of mitosis, it determines the width of the overlap zone in the central spindle. But if you knock that protein out, mitotic chromosome structure is also defective. That protein does everything, and so it's a kinesin motor that could make a loop because it's got 2 DNA binding sites. So, and Anton tells us that he doesn't understand what's going on with the high C that we see. So we have the cell lines that knock it out, and at the moment we haven't decided that we understand what's going on with the high C from the KIF4. So that could be, it could be that when you combine KIF4 onto the condensins, that you have now have a motor which maybe extrudes loop but has different properties.
Just a very fast follow-up. You were talking about basically now having the histones in the mitotic phase being responsible for this condensation. That's a very interesting idea. It appears to be a very expensive solution for condensation. You have to change all your histones, but appears very appealing. So, but it looks like that. Just have to generate all these proteins and replace all of them in order to.
But cells don't save money. Cells. I mean, look at actin polymerization, tubulin polymerization. These are dynamic polymers. Every time you do a subunit on or off, there's a GTP or an ATP that you just burn for nothing, right? I mean, cells burn a ton of energy.
Very interesting, very appealing, interesting story. Yeah.
Yeah.
Thank you.
Yamini, yeah. Hi, Bill. That was really very beautiful, especially the electron microscopy. Still remains some of your most beautiful work. So there are two sort of obvious questions here. You sort of talk about genomes being self-organizing when you're referring to histones being able to essentially push the chromosome loops that are being extruded into these kinds of very interesting shapes. But you left out...
I'm not sure I said that. I'm not sure I said that.
But the
I just said that I would be quite happy if the histone modifications were just making it do like that. I don't think it's making them do anything interesting.
So I'm curious in your mass spec if you didn't see phosphorylation of serine H1, because H1 phosphorylation is a known trigger for mitotic condensation. I'd be surprised if H1s had no role.
Yeah, H1 is the orphan now, right?
Right.
Nobody
Okay, so
there's what, 10 labs work on H1 now. H1 was it's the H1, for those of you who don't even know what H1 is the linker histone, which binds between, to the linker DNA, which we assume is between nucleosomes. H1 was the substrate that was used for mitotic kinases because H1 got phosphorylated in mitosis. And H1 I don't know.
So I'm just wondering if you're seeing, if Kumiko is seeing.
So a problem with H1 is there's 8 isoforms.
Yeah.
And they do different things, and they're complicated. So H1 is a little bit scary, and so people have kind of stayed away from it. But no, H1 is definitely sitting there, very interesting, and should be looked at.
Great. And then the second thing is, if you remember, there's this Conley-Ryder paper where they added RNase H, and the chromosomes explode. Mitotic chromosomes just literally explode. And I'm curious, in all these beautiful experiments, are you seeing any role at all for RNA as a structural element in these loop formations or loop maintenance? Thanks.
No, we haven't seen any role for RNA in loop formation. We know that RNA is involved in the chromosome periphery, which coats the chromosomes. And we had a few experiments that seemed to suggest that targeting RNase H to centromeres affected chromosome segregation, but those experiments were not followed up on.
So I was wondering if osmotic effects are involved in this condensation. You saw that, in the mitotic case, the density was a lot higher. So are there things like magnesium 2+ and things like that are involved in condensing this at different stages?
Well, I mean, Kazu Miyashima has published papers arguing that ATP regulates magnesium levels, and Tony Hyman has published papers which show ATP is an extremely interesting molecule, right? There's way more ATP in cells than you need for energy. And what Tony Hyman has published papers showing is that ATP can act as a molecule that can solubilize proteins that are becoming insoluble. I can't remember the name of, this is a particular class of chemical compounds that can solubilize bivalent, it's not Zwitteron. Anyway, ATP can do that, and actually we have confidence that it does that because I read that paper and I brought it up in a group meeting in Jayaprakash Arulanandam in my department, where people in his lab were trying to purify some proteins. They were crashing out, and they added ATP and they stayed in solution. So that's true. So these small molecules are probably having an effect, and nobody really talks about polyamines. Spermine and spermidine, which have 2, 3 positive charges on one chain and definitely can cross-link and bring negative compounds together.
So that should also change the cell volume and the nuclear volume also, right, as you go across the cell cycle. So is there...
Well, I guess if you're going to change the cell volume, you have to do things with water, don't you?
Yeah, because ions can change the osmotic state and then water can get pumped out, right?
Yeah, well, we haven't looked at that. Thomas Kramer, there are papers, if you're interested in that, look for Thomas. Thomas Kramer has put cells in hypotonic medium, and it causes chromatin to condense inside the cell. But it's not, that's not physiological, but it's you can do it.
Thank you. You mentioned the nucleosomes are incredibly sticky to the point that it basically becomes like a solid-like structure. The densities of the nucleosome inside mitotic chromosome sounds almost like a solid-like structure. And so I was wondering how inside a solid with these motors do you still get reorganization of the structure, because the diagonal does seem to move as the cell cycle progresses. So that sounds very counterintuitive in the sense, how are these small motors which are acting at piconewton organizing a solid that's almost kilopascal.
Well, Yamini is a chromatin geek. She can tell you this. I mean, this is one of the problems with working with chromatin. So if I make some nucleosome tinmers and I say, let's do some experiments, and we do some experiments at 5 millimolar salt, and now we say, oh, let's put them at physiological salt. They all crash out of solution. So it's one of the, chromatin is not soluble at physiological ionic strength. So why, what is it inside cells? Is it all gels, or is there something in there that's solubilizing it? I don't think, do we know the answers to these things, Yamini?
We know that I'm actually going to talk about this in my talk.
Okay.
They'll set it up beautifully.
Okay, wait for Yamini, and she will tell you an answer to that question. Thank you.
All right. Well, we are.